Good morning, everyone. Thank you for joining us today for this very interesting talk. I'm very happy to welcome Professor Ole Moritzen. Thank you, Professor Moritzen, for agreeing to give this talk. We are very grateful for your help, your cooperation in giving this talk at a very early morning time for you, but very convenient time for us. So thank you. As always, I request my colleagues who are on Zoom to hold your questions till the end of the talk. After that, please use the raise hand feature and we'll let you ask your questions. Our colleagues on YouTube, please type in your questions in the comment section and then we'll pick up those questions and ask them for you. I now request my colleague, Dr. Tripta Bhatia, to please introduce today's speaker.
Thank you, Prasad. And thanks, Ole, for joining us. So it's a very precious opportunity to connect with Professor Moritzen through science to initiate discussions about the meaning of key concepts such as green and sustainable transition using basic physics. So he is retired professor at the University of Copenhagen. He has a master's degree in physics and chemistry, PhD in physical chemistry, and DSc in computer simulation techniques. He has successfully made transitions between different research areas to work on interdisciplinary problems. Some of his most important scientific contributions include formulation of a quantitative and mechanistic model, so-called the matrix model, for lipid-protein interactions in membrane, together with Professor Meyer-Bloom, proposal and characterization of the liquid ordered phase in membranes, and its key importance for the understanding of fundamental physical properties and phase behavior of sterol lipid membrane system, together with his PhD student, Professor John Ipsum, a quantitative description of lipid domains in membranes as the physical basis for the membrane raft hypothesis in cell biology. And this was much before the raft hypothesis was proposed by Professor Kai Simons. Proposal, design, and constructions of liposomes for drug delivery in cancer therapy, theoretical description of the glutamate energy behind the molecular mechanism of the Umami receptor. Professor Moritzen also holds several positions, which includes National Danish Center for Test, Director and co-founder at National PhD School of Molecular Biophysics, President of the Danish Gastronomical Academy, the Director of the Center of Biomembrane Physics, Memphis, where I had the opportunity to work with him. I have experienced an interdisciplinary scientific environment created by him, where it was possible to combine theory with experiments and simulation. I learned that it is important to learn to differentiate between doing something for fame and something for giving back to the society. And building a professional trust among scientists is fundamental in science, something that connects all of us across the globe, and it reflects the gratitude for using the public money. He is the recipient of several awards for his work and outreach activities that includes writing books, talking to children, collaborating with professional innovative chefs, going to theater, meeting seaweed harvesters, researchers and medical doctors from all over the world. One of his long-term collaborators, Professor Martin Schuckerman, who is today here with us, says, Ole is an excellent collaborator who never fails to acknowledge the contributions made by other members of the team. His broad knowledge of physics in general and biophysics in particular, and his scientific intuition has helped to stimulate the creativity of those with whom he interacts. So with this, we all welcome you and we look forward to your talk. Thank you.
Thank you very much for the introduction, Dr. Bhatia, and thank you to the outreach team for inviting me to give this presentation. So I'm speaking to you from the other side of the world. I live in Denmark, but actually today I'm speaking to you from Germany, where I'm spending some time with my wife. What I'm going to do to you today is to give you some idea why a physicist may have gone through this crazy journey from something which is very fundamental and basic theoretical physics to something that is basically happening in our kitchens every day. And I was asked to give a little overview of what I've been doing and try to outline why I ended up where I've ended up now, with a keen interest in understanding what's happening in our kitchens and what is happening in our oral cavity when we eat, in particular when we eat together with other people. So I'll take you through some of the beginnings, what a physicist usually would do, working with systems of various complexity, and then take you towards what is called soft matter. And that is, from there, it's a small trip into food, because food is an example of soft pieces of material. And on the way, I've given a number of examples of what a physicist, or the way a physicist would work in order to work in these systems. So this is for the students. Why should one study physics? And the reason why physics is so powerful is that it deals with fundamental laws of nature, that is encapsulating the relevant variables out of very many different variables. And it can say something based on very simple laws of nature, say something about very complex phenomena. It's universal applicable whether you work with the universe as such, or something on an atomic or subatomic scale, on all lengths and time scales. So there's no system that can't be attacked by physics in some way or the other. And physics also is powerful because it's built on what we call the three pillars of science. It's theory, it's experiments, what you do in the lab, and it's also modeling and computer simulation, which in many respects have aspects of both theory and modeling as well as an experiment. Physics can give quantitative predictions about the world, and that also makes its power when it comes to applications in technology. So my own journey, which Dr. Bhatia asked me to tell you about, is that it started in sort of the borderlines of physics and physical chemistry in an area called statistical physics and thermodynamics. That's sort of the disciplines that deals with systems that are composed of many particles and how they behave as we change various conditions for the systems. And I've been particularly interested in using computers and computer simulation techniques to attack the problems that arise when you pose a problem in terms of statistical physics. And it's taking me all the way from something very esoteric nucleic magnetism, spin systems. I'll give you an example. And that brought me into what we call cooperative phenomena. That is the phenomena that persist in systems that are composed of very many particles. So we talk about Avogadro number of particles, and the various properties that derive from the cooperativity, such as phase transitions and critical phenomena. And that took me actually via studies on surfaces and various non-ordering processes into some more soft materials like liquid crystals and polymers, and eventually to lipid and lipid membranes, which I'll speak to you about shortly. And as examples of the work that we did here, and this is work with many different people. I don't have time to mention them as we come along, but that's work on cholesterol, what effect cholesterol has on membrane properties. It's about proteins and how proteins and their functions are affected by the membranes they're integrated in. And also an example of a technological use where one could use properties, particularly the physical properties of membranes, to design delivery systems, for instance, in cancer therapy. And the last part of my talk will be, and that's actually the bigger part, which is something that I'm very excited about now. That is how one can use the insight from physics, fundamental physics, to study taste and study texture of food, and how that can lead to food innovation. And my most recent work on the outreach field, which I'm very busy with now, that is seeing how one can actually use that to help humanity perform a green transition towards a more sustainable planet when it comes to the food systems. So let me start with the only equation I've got, which is also a very fundamental equation. It derives back to the 1920s, and it's an equation that describes how many different particles, we call them spins, you can think of them as small magnets, how they interact with each other in a system that is truly cooperative. It's called the Ising model. It's the name after Austrian physicist Ising. It was proposed by Lenz in 1920. And Ernst Ising solved the statistical mechanical problem of this system of spins sitting on the lattice. He solved in one dimension. It doesn't have a phase transition, but in 1944, Lars Onsager, a quite famous Norwegian theoretical physicist, solved the problem in 2 dimensions. Onsager was also Nobel laureate, not for this particular problem, but for the Onsager relations. And it was very difficult mathematical to solve, but it solved exactly in 2 dimensions. It's still resisting a solution in 3 dimensions. So if you want to have a go at it, please do so. So it just says that each spin interact with the neighbors with a product and a strength constant called J. And the solution of this in 2 and higher dimensions is that it has a low temperature, a magnetic phase, a ferromagnetic phase. And as you heat it up, and you can see the first graph and the graph in the middle, that's the magnetization, or we also call the order as a function of temperature. And you can see it decreases, and at a particular temperature called the critical point, critical temperature, it vanishes. And if you measure what we call response functions, they have peculiar properties right at the phase transition point. They blow up, they grow infinite. And for instance, the magnetic susceptibility, that is the way it responds to magnetic field, or the specific heat, the way it responds to changes in temperature, it has this weird behavior. And if you look at on the microscale, how it looks, you can see, and this is output from computer simulation on the bottom, of the very left-hand side, you have a black phase, all spins, let's say they're all pointing up. That's a ferromagnetic phase. There are a few excitations that are the white ones, that spin pointing down. And if you increase the temperature toward the critical point, you can see that the magnetization is vanishing. The sum of the upspins and downspins are getting closer to 0. And you see large patches of up and down spins. This is what we call the fluctuations. They're the critical fluctuations, and they become the source of the cooperative controlled organization of this 2-dimensional layer, something we can transfer to a lot of different systems, for instance, membranes. And then about the critical point, you can see that it's disordered, and you have random variations and spins up and down. So despite of the fact that this looks extreme, it's a very simple model. It's just a product of neighboring spins that enter in the unity function we call H, which is called the Hamiltonian. This has all the phenomenology of a many-body system, and that's why it's been probably the most powerful model at all in many-body physics. And if I plot it, let me see what happened here. Here we go. If we plot it again, this is the magnetization as a function of temperature. The critical point, it vanishes, and on the bottom I've written the characteristics of the behavior at the critical point. You have these so-called power law singularities, and in the 60s and 70s, there was a rush to figure out the universal aspects of these exponents, something I was quite interested in myself. And it actually led to a Nobel Prize for Ken Wilson in the beginning of the 80s, when he designed a theory to describe the universal behavior of this system. So even though this system model is for a magnet, we can use it for a lot of different things. If you look at the red arrow, there's an example of something we can bring now to membranes as the next thing. So imagine that you're at a temperature below the critical point, you have a certain magnetization, and then turn on a magnetic field. Then you can actually switch the magnetization such that it goes from up to down. When you have 0 field, it's symmetric, equally possible having a magnetic phase up and down, they're the same, but the field, you can switch it. And that switch, as you see on the left-hand side, that corresponds now to a transition where you have magnetization from one direction to the other. This is also a phase transition, which in contrast to the phase transition driven by temperature, this is what we call the discontinuous phase transition. There's a jump, in this case, in the order. So we can drive the transition from continuous transition to discontinuous transition. And this is something we can use to model a much more complex system, which is my next topic, and that is the behavior of membranes. So I'd like just you to keep in mind that we start with something very simple, and I will show you how you can use this for a much more complex system. All biological cells are bounded by membranes. And here we have a E. coli cell on the left-hand side, and the membrane core is a 5 nanometer thick layer of lipid molecules in which all the machinery of the cell sits. And on the right-hand side, you can see these big lumps there, the proteins, and a lot of other complex things. It's a sort of composites of polymers and lipids, and of course, the water all over the place. If we have a look at the lipids, there's a very great diversity in nature. They've made of what we call a head group, which is a polar part which interact positively with water, and then you have an oily part. This is where you have the fatty acid chains that are linked together, and we have thousands of different of these molecules in our cell membranes, but they share one unique common feature, and that is that they are amphiphilic. That is that in one end they interact favorably with water, and in the oily part they are dispelled from water. So it gives them sort of a mixed feeling about water. And this is the only thing you need to know about lipids in the following. But you can imagine now, if you expose these molecules to water, they have a conflict, because in one end they prefer to be close to water and the other not. And that leads to something which is extremely important for life sciences. I would say it's actually the most important phenomena in life sciences and formation of biological material. That is, you have molecules, in this case it's lipids, but it could also be proteins that are amphiphilic. So when exposed to water, they will organize to screen off the oily part, or possibly put in a more concise way, they'll try to maximize the entropy in the water binding network, and it leads to all these different structures. It could be a bilayer, as we saw before. It could be several bilayers inside each other. And such a single bilayer that sort of come back onto itself, it's called the liposome, but they're also what we call micelles, the little one on the left-hand side. If you put them in water, they'll form a monomolecular layer. So this is all formed by self-assembly. There are no strong chemical bonds. There are basically no chemical bonds. It's all controlled by the entropy of water and then the physical interactions between the molecules. I highlighted one of the objects here, the liposome in the middle, because you can now basically see this is a kind of a cell model, because it has an inside and outside. It can be used as a capsule to encapsulate, for instance, drugs, and this has been used for drug delivery. And it can be a very effective way to control the release of drugs. But it could also encapsulate other things. It could, for instance, be flavor agents that you want to release in your oral cavity. So this is a wonderful object. If we put a model of such a bilayer onto a computer, you can see how the self-organization happens. This is a very large scale calculation, and you can see the liposomes are being formed there, inside and outside. You don't see the water, because then you could not see anything. But this is happening. It's happening spontaneously. Now, coming back to phase transitions, lipid membranes also have phase transitions, and understanding these phase transitions and phase behavior are important for understanding how membranes organize, and hence also the organization of the membranes can control function. So here you can see how a physicist may imagine using the model I started with in the beginning, with basically two states of the molecule. So now here you have a piece of a membrane, where you can see on the left-hand side there's some order both of the chains and the molecules, and to the right-hand side it's more disordered. So one way one could model this is that for the ordered states you call them spin up, and for the disordered state you call it spin down. And in fact, you can modify the model, the Ising model I started with, really interpreting this state where you have the chains being straight out compared to the chains that are disordered by an internal magnetic field that can then turn the continuous phase transition, the Ising model, into an Ising model, which has a first order phase transition, which is indeed what you find experimentally for lipid membranes. So because of the fact that these molecules both can have positional degrees of freedom, they can sit a particular part of the membrane, but they can also, in the chain speed ordered or disordered, have really a mesophase description applicable to this system, such that the solid phase, spin-up phase, let's call it, has ordered chain, and the liquid phase, where you have lateral mobility, the chains are also disordered. We call the liquid disordered phase. So I need two labels to characterize the phase. So that's the extra level of complexity I need for a membrane. And just a little short while, you'll see why this is extremely important if you want to understand what cholesterol is doing to the membrane. Membranes have their share of cooperativity. You remember from the Ising model, there were singularities at the phase transition, and the same thing with membranes. And here, two functional aspects of a simple membrane in terms of its permeability, that it's the ligadness of ions, or its capacity to bind small ions, in this case ethanol. And you can see the underlying fluctuations, which I show images, is now reflected in functions that are extremely important for this membrane, where something leaks through or something actually binds to the membrane. So this is a system with just one component. If you now add more components, and those of you who studied physical chemistry or maybe material science will know that once you have more than one component, you add one more and one more, you get a very complex phase behavior. And here examples of mixtures of two different kinds of lipids. So we have binary systems, and it gives rise to a phase diagram where you have coexistence between phases. That's inside the loops, where you have coexistence of two types of phases, and it can be extremely complex when you have more phases. But considering that in a membrane you have thousands of different species, you can imagine that the phase behavior can be quite complex. So let me just show you an experimental figure of how one can actually image this using what is called atomic force microscopy. This is a binary layer. In fact, it's only a monolayer, so it's half a bilayer, but it's the same phenomenology you can see on the surface here. So this is a scan of a mixture of two different lipids, and they're characterized by two different chain lengths, and that is reflecting two different heights at the surface. And so you have, say, the long ones are the black, and the wide ones are the short. And in the little graph inside on the right-hand side, you can see the height differences reflected in the micrograph, and that corresponds to the change in height. So this is actually the source of something that Dr. Tripta has worked very strongly with. And one thing I need just to add to that in order to tell you what is important for regulating these phase coexistence and domains, that is cholesterol. And some of you may think of cholesterol as a molecule you should really avoid, but it's extremely important for the functioning of membranes. All plasma membranes have about 30 or 30 plus percent cholesterol if it's mammalian membranes. So it's the most dominant molecule actually in the membrane, and it's an amphiphile. It's integrated in the bilayer, and because of the fact that cholesterol has a sterile skeleton, it's rigid, so it's rigidified the chains, but at the same time it'll break the order among the molecules. And this is really the source of the importance of cholesterol in membranes. It's thickening the membranes, making them more stable, but it's also assuring that they're actually fluid. And the phase diagram for cholesterol with one lipid is here, and you can see it has quite complex phase behavior. And I'm not going to dwell with the phase behavior, but just look at the slide to the right-hand side where you see these domains, and on the bottom, the molecular dynamic simulations of a cross-section of the membrane. You can see there are regions it's highly heterogeneous, and there are regions where you have more of the blue molecules that are the cholesterol. It's thickening the membranes, so that you have these patches that are liquids, but they have ordered chains. And that's the way the liquid ordered phase looks, and it's a phase that is extremely important for membranes. And now you can imagine this is just the lipid part of the membrane. Once you introduce proteins, it's going to be more complex. And this is the model which I worked with my Bloom quite some years ago, but which is still a useful, very simple cartoon. And I show it to you here just to see how simple a model one can actually learn something useful for. So this is just a model where the protein is seen to be some kind of an integral particle that can couple to the thickness of the membrane. And the membrane can be thin or it can be thick, and the protein can be short, it can be long. And so that's a simple mechanic interactions. And that can actually be used to rationalize a lot of data on how proteins interact with membranes. And one of them is from one of Dr. Bhathia's work. It's a beautiful piece of work where she studied the most important membrane protein in our cells, that the sodium potassium pump, and how its function and also its position in the membrane is reflected by the way it interacts with the structure of the membrane. So this is a membrane that has 3 different types of lipids, that has the sort of phenomenology you want of a much more complex membrane, and then it has this important protein involved. And this is long story to describe what's happening, but we know now for sure that the way this protein functioned is intimately both reflecting and also impacting the lateral structure of the membrane. So that's what I wanted to tell you about soft materials, and now we'll go to food. So gastrophysics is a term that has come on the scene in the last maybe 15 years, and it composed of 2 words we know as gastronomy and as physics. And the way gastrophysics work, and it's very much inspired by soft matter physics, because soft matter physics deals with all these complex materials, whereas liquid crystals and polymers and lipids, and they all, many of them are derived from biological materials. And if you look at our food, it's all something that is biological origin, or something we have. Some may actually be alive when you eat it. Maybe a carrot is alive, or it has certainly been alive. So it's based on biological material. So it's the same soft matter we use in the kitchen as we can study in the lab from a more physical point of view. Gastrophysics we defined as a part of science which studies the part of soft matter which is gastronomically interesting. So it's not any kind of soft matter, and it's also not any kind of food, but it's something that has some gastronomic value, gastronomic interest in terms of texture, in terms of flavor. And maybe you can say that together with neurology, this science is it's the underpinning science of gastronomy. So it's different from cooking, although cooking is involved. It's different from food science, although it's involved. It's also different from nutritional and health science, and it's also different from sensory science, although it has some relation to this. So now you probably would say, this is very confusing. So what is it he's talking about? So let me give you an example. So one example could be raising this gastronomically, I would say gastronomically interesting question, at least it's been interesting to me. How can you make cucumbers? How can you make a preparation of cucumbers that are very crisp and crunchy? So once you said that, you could forget about gastronomy for a while and then study the cucumbers as a soft piece of material, take it to the lab and use all the techniques you've got available, all the experimental techniques here highlighted by microscope or theoretical calculations, and you can do various kind of experiments on the cucumbers and figure out how do you treat these to make the very crunchy cucumbers that may stay crunchy for weeks if that's what you want. It could also be other examples. I'll give you one just in a minute about cephalopods, that is mollusks that you want to have a particular texture. So some of my colleagues ask me, is that physics? And then I say, well, and going back to my example in the beginning, that physics is a very powerful and generic discipline. And physics, if I should be very bold, I would say physics is what physicists do. And what is very interesting with this kind of physics is that it brings in collaboration with another group of people, which you usually don't collaborate with physicists, and that's those who are very skilled in the kitchen, chefs, and a number of well-known schools around the world, universities, are actually having now programs where the interactions between chefs and scientists. For the last 7 years, I've been running a national center called a taste center, where there's people all the way from philosophy, neurology, sensory science, and also physicists, obviously people working in chef professions. We're trying to integrate and using gastrophysics as a new tool to work with food and also with programs directed to the general public and also to the educational system, throughout the educational system, using a combination of insights from many different disciplines. And what I'm going to tell you now is how we've used gastrophysics as a way of inspiring young people along with doing science. And I like to present with you an approach here, which is maybe contrary to what one usually says when scientists take on a duty to communicate about science to the public. And the usual model is that you do science and try to do the best science you can do, and then you pay back to society by communicating your results and communicating the use. But it's the research that comes first. What we've been trying to do is to do it the other way around. That is, instead of talking about research-based outreach, we talk about outreach-based research. And the way it works is that if you try to communicate to people about, in this case, about food, you actually sometimes come in the situation that you see there's something that is really unknown. You do not know it and others do not know it. There's a basis for doing some research, but it's inspired by your attempt to tell other people about a particular problem or solution of a problem. So the source of your inspiration comes from actually communicating. And this sounds a little bit upside down, but I'll encourage you to try to think about that. And I'm sure those of us who also teach would have a similar observation, that doing teaching, even the most simple course, as something you realize that maybe you don't quite understand all the depth, or there's something here that could really be interesting to study and hasn't been studied, but is inspired by your aspiration to describe the phenomenon to other people. And we found this very fruitful. So some of the examples I'm going to show you now that led to published research in very good international journals was actually inspired by talking to young people about the various aspects of food. So I'll give you some examples. So the examples I'm going to give you here is about taste, and it's about texture. I'll give you examples based on some, certainly in the Western world, some weird things that we don't eat much of, for instance, seaweeds, and also things like mollusks, cephalopods, squids, and octopus. And I'll indicate to you what we learned from these systems that can actually be used into a completely different other field. That is the field of plant-based food, which is a very important topic at the moment when we're going to enter a green transition. So let me start with telling you something I tell the young people, when I go around my country and speak at festivals and to schools, I tell them about how humans interact with food. And obviously we use senses. We have our five basic senses. So when we interact with the food, we both use our eyes, we use our nose, we use the palate, the tongue, we use our ears, we use the tactile sensation, the mouthfeel. So that's the way we interact with food, and that's the only way we can interact with food. And our perception of the food is then integrated in the brain together with all sorts of other experiences we've got, for instance, about social events. I'm sure you all know that a taste quite often is something we connect with an experience, for instance, being with our grandmother in a grandmother's kitchen or a birthday party or something like that. When it comes to the science part of interacting with the food, we have receptors. And I'll now focus on the receptors for taste proper. That is something that's happening in the taste buds, on the tongue. And we say we have 5 basic tastes, and they're all mediated by complex membrane-bound proteins, receptors that sits in the lipid membranes of our taste cells. And below there, you see cartoon of the 5 basic tastes and the receptors and some foodstuff that elicit these basic tastes. There's sour, there's umami, there's salty, sweet, and bitter. And one of the tastes I've been very interested in is what we call the 5th taste, umami, which I also now show you that it's extremely important to know about this taste in order to make green food delectable. So when it comes to food, obviously food is biological material. It's built of the same molecules as our bodies, and we basically have these 5 elementary particles. We have the carbohydrates, we have the proteins, we have the fats or oils, but then there's also 4th class, which we don't talk much about when it comes to food because it's not really important for nutrition, but extremely important for taste, and that's the nucleic acids. You can see in these cartoons, they're very big, complex molecules. And if you ask how do they taste, it's a bit surprising because they have no taste. These molecules are too big to be identified by receptors. So they have to be broken down into smaller pieces in order for us to appreciate their taste. So carbohydrates, they have to be broken down into sugars, such we can, and then they'll taste sweet. Proteins would have to be broken down into amino acids or peptides, and then they'll taste umami. Nucleic acids also would have to be broken down into nucleotides, and then they'll also taste umami. And the fats, surprisingly, have no taste of their own, but they're very important, of course, for providing for mouthfeel. There's one particular amino acid which is important for the following. That's glutamic acids, and it's called glutamate. And when it comes to nucleic acids, there are 3 nucleotides that are important for the following. That's inosinate, guanylate, and adenylate, which you'll find typically respectively in fish, inosinate, fish and meat, guanylate in fungus, and adenylate in shellfish and tomatoes. And it was discovered in 1908 that, and this is based on the study of a Japanese chemist, Kikune Ikeda, who studied the soup stock in the Japanese soup stock that is very rich in the glutamic acids, which derives from one of the components in this soup, which is seaweed. And he coined the word umami. And here you see the ingredients that goes into the soup. There's this seaweed, and then there's also fish product, katsuobushi, which turned out later to contain large amount of this inosinate, the nucleotide. And those 2 things together is what gives the strong umami, and we call umami synity. And the word umami was then coined, and it stayed with us since then, and it's now recognized as the 5th taste. And the wonderful thing about umami is that it's highly synergetic, such that if you combine seaweed as one side and fish on the other side, and if you're vegan, you use shiitake mushrooms, it has a very strong synergy, much more than the 2 components separate. And that's really the secret behind strong umami synergy. And now I'll bring you back to what we could then study in the lab, in this case, the computer, how to understand how this actually works. And so now I'll take you all the way down to the molecular level, understanding this umami synergy taste. So it's happening in the taste buds. It's a collection of cells. Each cell is bounded by a membrane, as we talked about before. And here we have a receptor. It's a big dimer protein molecule, which can bind in this motif. You see to the right-hand side, you can bind glutamate ion and it can bind a nucleotide. And then you have a signal which is transferred through the cell and eventually into the neural circuitry, and the brain says umami. And the way it works is this, and here you see sort of a biophysicist approach to taste. This is the part of the receptor that sits out in the saliva where the molecules that you detect with taste. So the left there's nothing bound, in the middle a glutamate bound, and to the right hand side you have both glutamate and a nucleotide, and you can see the dynamics sort of reflect the binding strength. And this is the molecular reason why you have this umami synergy. And if you think this is strange, you may look into your own kitchen and see how you actually combine different kinds of foodstuff. This you can download from the web, and it may be useful if you want to combine, because if you really want the maximum effect, you have to combine. And in the Japanese cuisine, which I'm very inspired of, it's a combination of seaweed and fish or shiitake. But in the British kitchen, it would be like eggs and bacon. It's the same pair of molecules that bring together. In the Italian, it'll be tomatoes and beef. Or Western soups could be contained of vegetables and meat, and you can then combine it. And recently, we published a paper which is actually, it almost went viral around the world because we took up a problem which sounds very extravagant, but the question is, why is it that some people think that champagne and oysters is a good match? And it actually comes down to the very same thing, that there's glutamate in one of the components and there are nucleotides in the other. In this case, it's oysters, and then you get the maximum effect. And I'm sure in your own kitchen you can find similar examples. So now I'm going to move a little faster here just to take you through towards the green transition, because one of the questions we could then ask as gastrophysicists, we can ask, how can we use this on materials that we have in the part of the world where you live? So we look for seaweeds in our part of the world, and we studied a whole range of different seaweeds in order to figure out where do we get maximum umami. And all this has come together in some disciplines we now call phytogastronomy, which is the gastronomy related to seaweeds. And using these seaweeds in the local waters can be very important for actually imparting umami to foodstuff that has very little umami. Let me show you some aspects of texture. So this is how the stuff feels in the mouth, which is extremely important. And here you can ask questions: how come that certain things feel the way they feel? And I'll show you examples of foodstuff like mayonnaise and butter. And we can use techniques, microscopy techniques, to actually look into the food and see how it's organized using basically kind of spectroscopy in the lab, where you can see which types of molecules you have at different parts of the foodstuff. And this is an example of a mayonnaise, which you will describe as creamy. It's composed of a lot of little droplets of oil in a matrix, watery matrix in the background. And you can always imagine that this must be creamy, because when you touch it, all these balls would just roll like ball bearings, and it'll feel creamy. So this is what physical chemists call an oil in water emulsion. If you turn it out inside out, where you have water in oil emulsion, it is what you find in margarine and butter. And we can also go to the cafe and ask how come that whipped cream or milk has a certain texture or feeling. And here you can see how you have a complex organization of milk fat, water, and air bubbles. That is the way a foam is is consisting of. So let me show you a couple of other examples, which is also gastrophysics, where we have taken questions from foodstuff and ask how can you actually make a desirable texture. I've been very interested in octopus and squid and cuttlefish, which is something we don't eat much in our part of the world, although they are in the waters. How come they sometimes are chewy? How can you actually change the structure and texture? And it's physically an interesting question because the muscles of these animals, which don't have a skeleton, it's highly cross-linked polymer network, and that is really the cause of all the difficulties. When you cook them, they can be chewy when you denature these proteins. But if you know a little bit more about the muscular structure, you can actually prepare them the way that they become tender, or at least have the texture you want. And once you set that and ask yourself the question, how can you make them with a particular texture, you can go to the lab and study them. I'll show you two examples of how one you can study them just by simple mechanics. You push on them, so do a strain-stress relationship. This is what we call texture analysis. And we can then relate the way we impact them or the way we feel them in the mouth with the way we treated them. We can also look at them in the microscope, or we can take it to powerful sources like X-rays, neutrons, and study how the collagen molecules, which is the source of the chewiness, how it's actually organized. And what you see here is actually a microscopy image. On the left-hand side, it's an uncooked squid, and on the right-hand side, it's a cooked one. Similar things you can use for vegetables. And now we're back to my example in the very beginning. How can you make crunchy vegetables? And here's some of the vegetables you'll see in Asia. And the secret is actually here you dry them first. And then you marinate them so they look quite tedious and dry and a bit not too pleasant when you see it on the left-hand side. But if you put them in a marinade, in particular marinade where they have divalent ions, calcium, you can cross-bind the pectin molecules in the cucumber, and then it can be extremely crisp. That you can also study in the lab. This is a texture analysis where you actually see reflection in the mechanics, which is actually what you hear in your ears. So here you impact with a certain force. If you study the force-distance relationship, you can see the force building up, and then it sort of goes into fracture, and there's a sort of a whole forest of fractures eventually before it completely breaks. And this is the signature of the crunch you hear in the mouth. So now you can study relationships between how you actually prepare the vegetables, different, say, cooking procedures, different marinades, different aspects of pH, different ions, and then relate it to a mouthfeel. And eventually you taste it yourself. You can also do microscopy and see what's actually happening in the cellular structure when you dehydrate and then rehydrate again, and based on the microscopy, get an understanding on what's actually happening on the small scale and how is that reflected in the way you feel it between the teeth. So let me finish with something I've been interested in and working on for the last couple of years. That is, how can you use the insight from physics, in particular gastrophysics, the knowledge of the physics of soft materials, and how their texture and how their taste gives rise to different kinds of pleasures when we eat them. And that brings us to the big question at the moment, that is, how can the world go more green? How can we provide for a more sustainable food system where we have a much more plant-based foodstuff? And of course, this is much more an issue in the Western world, where we eat much more meat than you do, those of you who listen to me from India, where you have a culture where you actually have a very large part of the diet being based on plants. But this is not true everywhere in the world, and it's a big problem that we eat too much meat and too little, say, vegetables and greens, and we eat too high in the food web. So we have to read further down the food web where we find the plants, the fungi, and also the algae. It could be the macroalgae like the seaweeds. And the question and the challenge that we now face is that for those countries where people eat a lot of meat and they look at all these vegetables, beautiful vegetables, and they say, well, we like to eat more vegetables, but they're not really, they don't appeal to our palate. And this is where the science can come in as a help, because there's no need. People know for sure that this foodstuff is healthy. We also know that it's more sustainable than eating high up in the food chain. But the reason why it's not eaten is that it doesn't have the appeal to the palate, and there are biological reasons for that, and that is that plants do not have umami except for some of the ripe fruits. So the green parts do not have umami, and the reason for that is that plants don't have muscles, and they don't have these free amino acids that leads to the umami taste. And on the other hand, also most of them are not sweet. Again, and that's of course because the plants do not want to be eaten. They have all the sugars bound in carbohydrates, and they're also generally bitter in order to deterring us for eating them. So if you confront that with the universal craving for humans, and this is part of our evolutionary path that we evolved as meat eaters 2 million years ago, we're craving the particular taste that is characteristic of meat, and that is umami. So it's uphill eating more green because they lack umami, which we evolutionary are primed to eat. And they also lack, obviously, sweetness and have bitterness. So what do we actually do? So this is where gastrophysics can be helpful, combined with actual gastronomy, and that is figure out how to actually add umami. And you can do that with small amounts of meat, fish, and dairy as well, eggs, fungi and algae. Or you can go the other way, and that is releasing the green stuff's own potential for becoming both sweet and umami. And when I say potential, it's because of obviously plants. Think, for instance, of beans and linses. They do have a lot of carbohydrates, but they're not sweet because the sugars abound in carbohydrates. But by fermentation, we can release the carbohydrates, and they turn sugar. They turn into sugars and hence sweet. The same thing with proteins. Lots of proteins in many different kinds of plant material, but taste, it's hidden in the amino acids and small peptides. They have to be released, and we can also do that by fermentation. And altogether, while we're working on the taste aspects of the plants, we also have to care for their texture. And this is, of course, something that chefs and those who cook, they know a lot about this. But I would claim that learning from science, we can find other routes to do this, in particular, routes that could help many more people eat green, because we talk about an enormous increase in world populations towards 2050, almost 9 billion people have to be fed. And the current food systems of the world, they're not sustainable. There's something that has to be done. And in order to eat from further down the food web, in order to eat more plants, I would claim that science can help us. And based on some of the insights I gave to you, it's my belief that also physicists can contribute to the green transition. And it has to happen together in collaboration with a whole lot of other different disciplines, and not least those people who are also in the crafts, working with the crafts that is the chefs. So I'd like to thank you for your attention. I ran a little bit over time, but there's still time for questions and reflections. And I put on the last slide with some of the books I've been writing, where you can, which have been translated into English. and the one on vegetables, the one on the Japanese vegetables, as will be coming out just in a few weeks. And towards the very low bottom, there's a book about lipids, which is a very simple-minded book, which can be used as an introduction to understand how fats and lipids are extremely important for life, not only as a part of the way we eat, but also the way all organisms are built. So thank you very much for your attention, and I'm prepared to answer whatever questions you may have. Thank you.
Thank you, Professor Mauritsen. It was a fascinating talk. And for the audience, it's now time for questions. I already see there is a question in the chat.
Hello, I'm Madhusudana from Bangalore.
Yes, please.
I'm a vegetarian, so I don't even know what umami tastes like. But we always add a lot of spices in Indian vegetarian food. Do you think spices have anything to do with this umami taste?
The only spices that I can sort of see scientifically would have some umami are those that are where you have some kind of fermentation involved. But spices in my universe are of course wonderful to use in the vegetarian cuisine. But spices are mostly aromas and not taste. And actually, when Dr. Bhatia and also some of other Indian colleagues were in my lab over the years, we discussed this at length. Where do vegetarian cultures, where do they get their umami from? Because we all have the same genetic material. We all have the craving for umami. So we need it in some way or another. And one way you will find it in the vegetarian cuisine is in tomatoes. And the most powerful way to get it in is actually via fungi, I mean mushrooms of different kinds. So this is usually how vegetarians, and in particular vegans, how they deal with their craving for umami. It's not the spices, unless you have the fermented spices, because spices are all for the nose. It's
no. What about jalapeno? I thought it was a taste, not just for your nose. Jalapeno. You know, jalapeno.
Jalapeno peppers.
Oh, peppers.
They are not just for the nose. I thought they were quite.
Yeah, but the jalapenos and other from the capsicum family, that's the capsaicins, and that's chemostatic action. That's the irritation, the warming, which is of course also a very important part of a dish and a meal. And it's not. I had a little section of that because we actually measured these capsaicins, also inspired by being gastrophysicists. We asked the question, how strong can these chilies be from climates, cold climates, which is the Danish climate? And but that's not taste. It's chemostatic action. It's irritation, which is of course also very important for a meal, but it's not umami.
Okay. Thank you. I think Professor Ajay Sood has a question.
Hi, I really enjoyed the last part of your talk, which is so very fascinating. So I have two small queries, curiosities rather. One, is it possible that if you take vegetables and by genetic modification or by breeding, they can produce umami? Is that a possibility?
I'm not a biologist, so I can't say. I don't know for sure, but I say why not? Because and I think it is a matter of those who work with breeding, that they focus on certain aspects. And there's an example of what has happened the other way around. Certainly in our part of the world, tomatoes are almost completely uninteresting to eat because the breeders, they are focused on fast growth, high volume, uniformity. And during that process, all the genes that code for aroma compounds, many of them are gone. So the tomatoes are not like tomatoes used to be. I don't see why one could not work it the other way around. But it's still, I know for biologists, a big mystery why certain ingredients like seaweed, how come they have large amounts of free amino acids? And I don't have the answer to that. I don't see why it shouldn't be possible for breeding or genetic modification to produce plants that have more umami. And I think that's what I've been telling you today. If that could inspire some to think about in these lines, because I feel strongly that the reason why many people from the West, they have great difficulty cutting down on the meat, that's because they do not get the satisfaction which is due to the umami taste. And it is, if that's the case, there's a very simple cure how to deal with it. And of course, vegetarian cultures have found out a long time ago. And one route is, as I mentioned, fermentation. Another route is used to use condiments that add this little umami you need, where it's a drop of fish sauce or some soy sauce, various kind of fermented, usually it's pulses, fermented pulses that have a lot of proteins that can lead to amino acids. So there are ways, and I think if those who feel that need meat, if they don't confront themselves and realize the reason why I have difficulty eating green stuff, even though all recommendations is that we should eat more plants, it has to do with the biology, or it has to do with our genes, and so we have to face the fact that we crave umami, but there are ways to get it without eating meat, without eating a lot of meat. In fact, you can do with very little. And my vision for the future here is that it's unlikely that the whole world population will go vegetarian, and certainly not vegan. It's not desirable, but with a little help from the animal kingdom, you can get all the way.
My small query was: is there a role of cryo freezing or cryo drying in this conversion of the food to some texture and so on? Because somehow you didn't touch it, but is there a role of it?
I don't think it'll have much effect on the taste, but it could have on the taste experience in the sense that it changes other properties. So certainly it'll change the texture. And I think also texture. This is why has been very interested in the Japanese way of preparing vegetables to make them crunchy. I think texture. I noticed that with children in particular, certainly the children in our food culture. One of the reasons we see they don't eat vegetables is that the vegetables are cooked to death. I mean, they're soft. They're not pleasant. Children, and I think many adults as well, they want vegetables that have some crunch, some interesting mouthfeel. So that is, of course, a matter of physical chemistry, understanding how you change the texture by the way you the heat treatment or freezing, if that you mentioned that, or by the way you store it in different kind of paste or marinades. It's solvable.
Thank you.
Thank you, Professor Sood. Professor Sriram Ramaswamy has a question.
Hi. Yeah. So, I mean, part of my question was answered. In the South Indian tradition, 6 tastes are recognized. 4 being the ones, 4 of the 5 that you mentioned, not umami, but also hot, spicy, hot, and astringency is officially listed as a taste. You think you?
Yes.
So, I mean, what's the molecular basis for these? You said spicy is a nerve reaction, not a taste bud reaction.
Yes.
And astringency is a mechanical reaction, or what is it?
Astringency is something that's happening to the protein in your saliva. Mm-hmm. If you have the proteins will bind to typically phenols or polyphenols in the food, and it will make on a small scale a kind of a clumping such that it doesn't lubricate.
I see.
That's why you feel it's sort of astringent or it's dryish. And of course, one can call. You're welcome to call. I do the same. We call it all taste. And in fact, it has a lot to do with the language. For instance, in my language, we don't have a word for flavor. Mm-hmm. That is sort of combining both taste and olfactory sensation, also mouthfeel. And some even say that flavor also includes what the eyes see and what your ears hear. And so I think it's perfectly respectable to talk about astringency and also chemostatic action as you have from chili as
Right.
taste. But the physiologists would insist on, before it's a taste proper, you would need to have some identified receptor and a mechanism, neural mechanism known. So taste is then sort of something that is happening in the taste buds or in the oral cavity. And there are no reasons to believe that there should only be 5 basic tastes. It's not
Right,
like,
sure.
I mean, defining a basic
Sure,
taste is
sure.
not like finding an element to add to
Right.
the periodic table. So some now discuss whether there's actually a taste of fat, but we can actually, there's some indication that we can taste small chain fatty acids.
I see.
And there's other, but it's complicated because there are all sorts of interactions between the different tastes. And there's a new word on the scene, again, a Japanese word called kukumi, which is describing sort of a continuity of the taste experience, something which is very prominent with garlic. And in fact, garlic, and that may also be interesting in context of Indian cuisines, that using garlic can actually enhance other tastes, such that if you have just a little bit of umami, you can actually buy garlic as well. There's also other various kind of fermented products that would also wire this kukumi action, which has some neurological basis, but not in detail, but it can enhance a little umami. And that is actually also important for another point of view, because parts of the world suffer from huge problems with obesity, because we have cheap products which has a lot of carbohydrates, and we crave these carbohydrates, and we want to cut down on sugar. But it's known for almost 100 years that if you know how to dial on umami, you can actually perceive something as more sweet without having more sugar. So one of the chefs I work with, he actually made a dessert, an ice cream with 30% less sugar, but adding a little bit of tomato juice at the inside of a tomato. You couldn't taste the tomato, but it has just as much sweetness as a dessert with more sugar. And a similar effect, a cross-modal interaction or or interaction with other basic tastes, and that's salt. We know umami can enhance saltiness. You can almost cut down 50%. And that's also good news because hypertension is also getting a huge epidemic problem across the world, and we use too much salt because salt is enhancing other flavors. But umami can actually help you feeling more. The perception of salt is higher with less salt.
I see. Okay, thank you.
And there are a lot of things we don't understand, but it's all in the brain.
Sure. So I have one tiny additional question. When you talked about fermentation, again, there's a large number of fermented foods, grain and lentil-based fermented foods. Does that fermentation also produce umami, or is it only?
Yes, it does.
I see. So yes, I see.
Yes.
Okay. Thanks.
Vibhuranjan Sarangi.
Yeah, thanks for a very fascinating talk. So I have a very curious question. Is that suppose you have two kind of mixed flavor in a particular food? I mean, is there our body does it have an affinity for a particular taste? I mean, is there a dominant variety of taste so as to say in this 5 kind of taste?
Yeah. When it comes to taste proper, that is something that is combined of the 5 basic tastes, and any taste by definition is a combination of the 5 different tastes. And now we're forgetting about the nosy aroma. And then we would say we can distinguish maybe some thousands different kind of combinations, but it's quite different once you couple in the nose, because you have many more, maybe trillions of different ways of, and we have also many more receptors, olfactory receptors. And when it comes to preference, there are a lot of other things we have to take into consideration, because that's culture, that's ethnicity, it's what you were brought up in, with whom you eat, family. But when it comes to taste proper, that is on the tongue, there's something, this is very fundamental also for understanding how to eat more green. And that is that all of us, of course there can be genetic variation, but all of us crave umami and sweet. And this is, there's an evolutionary reason for that, and that is that sweet is a signal of calories, it's good for survival. Umami is a signal of proteins and easy accessible proteins, it's also good for us. So those two tastes we are all born to like. You'll find it in mother's milk. Mother's milk is sweet because of milk sugar, and it also has free amino acids. So we crave those, whereas something like bitter and salt and sour is in between. And certainly we detest bitter from young. We learn it gradually. Now, that's different from preference, because preference is something you and I, and we can discuss this wherever. Some like certain taste and flavors, and mostly flavors, and other things. So that's something we can't really, it's more difficult to make science out because we are individual, and it changes also during life, and it changes through from young age to older. But taste proper, in particular sweet and umami, that's something we crave. And that's why I think it's so fundamental when we talk about eating more plant-based food, because that's something we crave, and it's something we may miss in a certain type of foodstuff.
Thank you so much.
Okay. Here's another question from Uddalo, and he's put it on the chat. His question is more of history, culture of food kind of a question. Here goes the question. Does the invasion of a foreign umami source indicate that the local food culture lacks it? For example, despite India having alternate souring agents, we still use tomatoes. Tomatoes are supposed to have come to India fairly late. So that's his question.
Yeah. I'm sure as different food cultures will adapt things from outside. Usually it's a slow process. It's not something that's happening very quickly. We don't change food culture that quickly. Certainly tomato is maybe the most prominent example of something that leads to umami. And I think also coming back to what I said before, that it's something primordial with us liking umami. The tomato is actually one of the foodstuff that is liked all around the world, and I think that's the reason why it is. And it has to be ripe tomatoes, by the way. And also scientists some time ago, they also studied the umami potential, comparing the outside of tomato and the pulp. And actually the pulp has 3 times as high intensity as the outer part of the tomato. And the tomato is also unique in the sense that when I spoke about umami synody, that is, you needed both glutamate and you needed nucleotides. The tomato is singular, interesting here, because it has both. So it can provide umami synody without being paired. As for sour, I'm not sure I understood the depth of the question, but sour is something we learn how to appreciate, but we need a certain amount and not too much, and that's of course to regulate our bodily function. The same with salt, by the way. It's something that we sort of also regulate to some extent. Certainly in a native food culture. Did this answer the question, or?
Yeah, I think it's also related to the second question that he has, which is, does processed milk like yogurt have any umami in it? Because historically it's been said that some time ago in India, the chief souring agent in the food was curd, but later on it switched over to tomatoes. So I guess that's how the two questions are related.
To the extent that the souring has involved the lactic bacteria, it would have produced umami compounds. And in fact, cow's milk is one of the most interesting examples of showing how umami comes about, because fresh cow milk has very little free glutamate. Whereas if you take the most mature cheeses, and the Parmesan cheese is the one that is known to have the most. It has 3,000 times higher concentration of free glutamate. So it's happening both during the fermentation process and the aging process that the proteins are broken down. And that's also why I would advocate for the green transition that we should use dairy. I mean, some people now say, well, we have to throw dairy out because we can't use animals. But I think just a little bit of milk products and just a little bit of mature cheese or fermented milk will bring something to the vegetables. So I think that kind of dairy is important for promoting a green transition.
So we have one question from our colleague who is watching this on YouTube. The question is there a parallel story for carbohydrates? Texture, taste versus internal structure? And how do you control these two if it is true for carbohydrates?
Could you please repeat the question?
Yeah. So the question is there a parallel story for carbohydrates in terms of texture, taste, and internal structure?
Yes. I think most people would know it from starch, which is a carbohydrate. And if you think about how you make bread, where texture is really extremely important thing, both for protein, but also carbohydrates and the gelatination of the carbohydrates when you have a moist dough. The other example, I mentioned one example is also carbohydrates, and that's pectins. Pectins are these soluble fibers, carbohydrates that bind cells together in plant tissue, and the strength by which they bind the cells is determining the texture of the plant. And in the kitchen, you would regulate that by mostly, to some extent, pH, but mostly via ions. So that they had a strong effect on texture of by the binding of pectins, the texture and the strength of the cell bound, and hence also the way you will feel it in the mouth. And there's a whole range of, most people don't realize, but during the food, a lot of processed food, the industry and also in our kitchens, we use all sorts of different kind of carbohydrates as dilation and thickening agents. Quite often, say in milk products, most people don't realize that in commercial yogurt, you have carbohydrates, some of them actually extracted from seaweeds, carrageenans. It's a technologically extremely important part of all these complex long chain molecules that form complex structures that changes viscosity, say, and even whether it's a rigid gel or not. So think about it in your daily life. They're all over, and they're natural compounds, and they're extremely important for keeping sort of the foodstuff in some kind of shape. Think about ketchup. I mean, it's also a carbohydrate that in commercial added to commercial ketchup. It's called xanthan gum, which is a complex carbohydrate that turns the ketchup into a fluid that doesn't drip, and actually has this very interesting non-Newtonian behavior, which is known as the ketchup effect, that it flows with different velocities depending on the shear forces you exert on the food. Ketchup is actually an interesting lab to learn about the physics of food.
Yeah. In the interest of time, maybe I'll take one more question, and that question comes from Mahua. Mahua wants to know whether the process of cooking, both the procedure or the duration, have any effect on umami.
heat itself for a short while does not. And in fact, we've studied that during this process called sous vide, which is a prominent thing nowadays. For instance, with meat, it doesn't change. It may change the distribution of where you have glutamate, whether you have it in the fluid or in the tissue. But it has to be more. I mean, in order to break down the proteins, you have. I mean, enzymes that's the best way of doing it. But also, obviously, higher temperature can break down the proteins. But fermentation and aging, that is the most effective way.
I guess there are no other questions. I think we have answered all of them. I would like to thank Professor Moritzen for this fantastic talk and for interacting with us. Very early for you, but still you took time out for us. Thank you so much. I would also like to thank all the people who have joined us from as far as Vancouver, again in very ungodly hours. Thank you so much for your interest. Thank you for joining us. I would like to thank my colleagues, Amit, Tripta, and Mahua, who have been working behind the scenes to bring you this talk. Thank you all. Thank you so much. I'll see you again in the next talk. Thank you.
Thank you.
Thank you. Thank you very much. So should we leave then now?